So now we have our second speaker. His name is Haider Inem. He just became a doctor through the Penn State University in the Pitcher Lab, and his thesis was based on developing technologies for correcting error in sequencing, and he's using a data set of deep mutational scanning variants to look for variants of the BCR-ABL gene that is related to leukemia. So he wants to be able to call these variants up properly without error, to really measure depletion and which ones are enriched. To the final goal here is looking at drug resistance for chemotherapy, for cancer therapies. So thank you, Haider, for coming, and go ahead.
Thank you so much, Edrine. I appreciate the kind intro, and I feel like you covered my first slide, so I don't need to give my intro anymore, which is great. Can you guys hear me just fine? Just to verify.
Yep, all good.
Okay, sounds good. Okay, so good afternoon or morning, wherever you guys are at, and thanks for hosting me. As Edrine said, I recently defended my PhD in Justin Pritchard's lab in biomedical engineering at Penn State. And today we're going to be focusing on using DMS, deep mutational scanning, to address some of the pressing needs in drug resistance research in the context of targeted anti-cancer therapeutics. So I'll start off with a brief background on what targeted anti-cancer therapies are. These are therapies that are drugs that have been rationally designed to inhibit driver oncogenes in cancers like a gastrointestinal stromal tumor, lung cancer, and in leukemias. So the first targeted therapeutic, imatinib, shown here in red, directly and specifically binds and inhibits the genetic fusion gene BCR-ABL, which is a chromosomal translocation and is implicated in CML, which is chronic myelogenous leukemia. And over the last 25 years, imatinib therapy has dramatically improved the survival of CML patients over chemotherapy alone. However, as you can see in this Kaplan-Meier graph, the patients undergoing imatinib therapy still do relapse a lot of times. And the primary mechanism of resistance is point mutations that reduce imatinib binding to BCR-ABL. And these point mutations are also the primary culprit behind the failure of various other targeted therapeutics. So they're important to study. And just like in inherited disease genetics, there are rare variants here that are consequential for patient survival. So let's see how this plays out in the clinic. In this graphic, we can see that patients with a sensitive population of cancer cells undergo an objective response to therapy, to imatinib therapy, to TKI therapy, but resistance cancer cells eventually do grow out and cause the patient to relapse. Moreover, different patients relapse with different mutants. And finally, there are various drug options that clinicians can use to treat these relapsing patients. Rare drug resistance variants make the picture even more complicated. So we will focus on the rare nature of drug-resistant variants. And we'll focus on our attention of imatinib as a case study because it's the frontline therapy for CML, the leukemia that I'm talking about. And we have 20 years of clinical data about imatinib resistance. And so far, over a dozen mutants have been characterized to cause imatinib resistance. This means that the resistance phenotype has been well studied in vitro studies. However, if we zoom out of this histogram and we look at the complete picture, we see that there are a lot of variants that are seen in the clinic, more than 500, in fact. And in fact, these variants collectively take up more than a third of the imatinib relapse patients. So we coined the term variants of uncertain drug resistance against imatinib to describe these rare variants. For imatinib, we have more than 20 years of clinical data, but for other tyrosine kinase inhibitors of ABL, the picture is understandably much less clear. Most of the variants are variants of uncertain drug resistance. So you obviously can't run clinical trials powered to test hypotheses for rare variants, right? So in the absence of clinical resistance data, any source of information is critical. And this is an important question because when you detect a rare variant upon relapse, clinicians have to decide what course of therapy to take. It's tough because in some cases, the variant just might just be an inconsequential bystander and is not actually driving relapse. It's not responsible for driving relapse. However, when it is driving relapse, clinicians have two potential courses of action. They can either escalate the dose of the current drug, and this would require an understanding of the clinical pharmacology of the current drug, or they could switch over to another drug altogether, and this would require an understanding of the resistance vulnerabilities of the new drug. To help geneticists and clinicians navigate these decisions, the National Comprehensive Cancer Network makes treatment recommendations, sometimes just based on in vitro data when clinical information is so limited. As you can see here, the NCCN is only able to cover the top few resistance vulnerabilities of a drug because drug resistance data for these rare variants is so sparse. So we can use, so the question we asked is, can we use DMS screens to quantify drug resistance and help guide some of these clinical treatment decision making process? Just a quick note here, the FDA in the past has approved drugs to be used against rare variants based on just in vitro evidence. And here are two examples. One is in CFTR and one is a targeted therapy against PDGFR alpha mutants. This targeted therapy is known as Avokit. And the label, if you really focus on the label, you'll see that it's approved at 300 milligrams of daily use for certain PDGFR alpha mutants. So the point is that before we can make any treatment, clinical treatment decisions based on our DMS screens, we need to have a good understanding of how to relate our in vitro data to in vivo systems. And estimating the effective concentration of drug in the serum requires an understanding of the pharmacokinetic profile of the drug. And by that, I just mean mostly measurements of drug concentrations taken from patients' serum over time. And to tackle this challenge, my colleague Scott Lehau has built classifier models that translate between in vivo drug exposures and in vitro screen data. And these measurements are used for gold standard mutants, known resistant mutants that we've studied in the lab, but they can also be generalized to test sets of other mutants. To train these models, though, you need dose response data at multiple doses. However, and when you look at the way we typically study dose response relationships in lab, it's done on a clonal basis, right? We give clonal populations of cells increasing concentration of a drug, and these are called IC50 studies because the aim is to identify the concentration at which 50% of the population dies off. And for a sensitive population, that number would be low. For a resistant population, that number would be high. I want you to note how resistance is not a binary phenomenon here. It exists on a spectrum. So a mutant that looks resistant at a certain concentration might look sensitive at a slightly higher dose. So PKPD, pharmacokinetics, and pharmacodynamics really matter here. And we need to know not just whether a mutant is resistant, but what dose of drug would potentially sensitize that mutant. The current pool screens, however, usually are run for drug resistance, at least, on a single dose or on just a few doses. And so far, we know that to really have a clinically meaningful impact, a DMS screen measuring variants of uncertain drug resistance would first need to be run at multiple clinically relevant concentrations, and second, be able to tie back the in vitro concentrations, in vitro data to in vivo drug exposures. So when we sought to build a DMS screen for drug resistance, we addressed some key technical barriers to getting good estimates of drug resistance. So the first two here are related to sequencing error rates, and the last two here are related to measuring drug resistance well. The first barrier that we addressed was error rates. In DMS screens, sometimes you have tens of thousands of variants at allele frequencies down to 1 in 100,000. And some well-published studies have used regular NGS to screen and track drug resistance variants. And a lot of times the data from the literature, we're looking at P53 drug resistance screens, and the noise from the unintended codons is almost as great as the signal from the intended codons. I want you to look at how all possible unintended codons in this particular screen were detected in this screen. So you're detecting all codons that you're trying to make, but also everything that you're not trying to make, which goes on to say how much error there is in this data. So we see this in the literature, and we see also this in our lab, where if we try to mutagenize ABL in the blue region, but we're not trying to mutagenize it in the red region. If we just use NGS, the signal to noise is very low here. So to address these sequencing error rates, we used duplex sequencing, which I won't go into too much details here, but it's a tag clustering based error correction approach that works by appending molecular barcodes to shared pieces of genomic DNA. And we can use these barcodes to count mutations with pretty high confidence. So duplex sequencing has an error rate of 1 in 100 million, and NGS is down to 1 in 1,000. These UMI barcodes, these single strand barcodes, have an error rate of about 1 in 100,000. So when we use duplex sequencing, we see a vastly improved signal to noise for drug resistance in these screens. Also, the one thing I wanted to mention here is duplex sequencing appends molecular barcodes to genomic DNA. So it's very adaptable in which applications you can use it in. We are currently using it in, instead of sequencing sgRNAs in base editor screens, we're using duplex sequencing to directly measure mutant pools at very low allele frequencies. Yeah, but duplex sequencing isn't optimized for DMS. So what we realize is if we use a regular duplex sequencing kit and the depths afforded by it, 20,000 depths on target, only allow us to see 2/3 of our library. And you can see another way to visualize this is if you were able to see the baseline completely at day 0. After you treat with drug, the problem is exacerbated because the more resistant variants do take over the population, driving the less resistant variant or the sensitized variants down to very low allele frequencies. But we adapted an approach that enriches for not just all genomic DNA, but targeted genomic DNA. So it's a targeted way of appending molecular barcodes. It uses CRISPR to cut out pieces of genomic DNA that you're actually interested in sequencing. And by using this CRISPR-DS approach, by scaling it up, we were able to get duplex depths of 10-fold greater depths, so 300,000. And when you use these depths, we were able to see not instead of 2/3 of our library, most of our library using standard duplex depths. So having addressed the technical barriers of error rates and low duplex depths, then we focused our attention on measuring drug resistance well. So one of the first things that we did is that we did a spike-in experiment where we took the 17 most clinically abundant imatinib-resistant mutants for CML patients, and we measured their enrichment for imatinib resistance in vitro at low allele frequencies of 1 in 15,000. And when we measured the enrichment of allele frequencies, we saw significant depletion artifacts in our screens. And these happen when the most resistant mutant quickly dominates the entire population, and the less resistant mutants that are still resistant look like they're actually depleting of the population when they're actually not. So the predicted trajectory here is drawn from clonal dose response studies. We individually made these cell lines and studied their IC50 curve and drew out this predicted trajectory. And the measured trajectory is what we see when we spike them in at very low allele frequencies. However, we came up with a way to convert allele frequencies to net growth rates, and that removed these depletion artifacts. And all mutants seem to be growing at their expected net growth rates and their expected trajectories. So, having addressed the technical barrier of depletion artifacts, we next the final problem we had to address is dose response variation. So when we plot out dose response curves, these IC50 curves, typically people plot out the mean trajectory. However, if you look at the replicate to replicates differences, you can see significant day-to-day deviations in growth rates that are characteristic of slight differences in dose applied across the replicate. And we saw these dose response variations in our 17 mutant spike-in assay as well. As you can see, one of the independent replicates looks like it's a little more fit than the other, and it's presumably treated at a little, it's responding to drug in a little more fit of a manner. So we asked the question, can we correct for these dose response variations? And to correct for them, we built an inference-based algorithm that first infers the difference in dose applied across the replicates and estimates what the growth rates of the replicate would have been had both replicates have been treated at the exact same dose. And this approach was able to improve our mean square error across replicates by about 10-fold. So having addressed some of the drug resistance specific technical barriers that we needed to address before we could measure resistance well and be able to relate them to clinical decision making, we scaled our screens up. We took all 252 amino acids in ABL, and we mutagenized them with the help of Twist Biosciences. And the library that we got, we transduced it into BAF3 cell lines, which are a murine lymphoma cell line. And we treated these cell lines, these libraries, at multiple concentrations of imatinib. And this is just one of the heat maps that we got, and there's a heat map like this for every dose that we tested. And with our screen data, we saw very good correlations across our replicates. And furthermore, all known imatinib-resistant mutants significantly enriched in our screens. So it was a good sanity check that our method was able to quantify the resistance well. But what about a variance of uncertain drug resistance, which is the focus of this talk? We focused our attention on these VUDRs detected in treatment relapsed patients, so imatinib relapse patients. We looked at the catalog of somatic mutations in cancer, which is a data repository run by the Sanger Institute, and it gave us treatment data from about 3,000 CML patients that had relapsed on imatinib therapy. In total, there were about 107 unique variants of uncertain drug resistance, and we saw that about 18 of them, roughly 20%, were strongly imatinib resistant. So the rest of them were actually they seem like they're actually not driving imatinib resistance, and they're bystanders of another resistance mechanism. For the 18 variants that do drive relapse, though, the question becomes, what do we do next? Do we just use the same drug and dose escalate, or do we switch over to another drug? So we tried to address that question. So to give you some more context, 400 milligrams of imatinib is the approved dose for frontline use, and it has been the standard dose. And it gives you an average serum concentration of about 300 nanomolar. 800 milligrams has been clinically evaluated. And when we tested whether 800 milligrams would be efficacious against variants that are resistant at 400 milligrams, we saw that 7 of the 18 mutants were sensitized upon treating with a higher concentration of imatinib. So dose escalation is a viable therapeutic strategy for a subset of these variants of uncertain drug resistance. For the rest of these mutants, the question becomes, what's the next optimal drug? And this is where we head into the future steps of this project. And so, because so far I've developed the first scalable duplex sequencing workflow to really quantitatively look at imatinib resistance in ABL. And in the near future, we are characterizing other drugs with this workflow as well. This is some of the work led by Josh Reynolds and Marta, who are our in-house duplex pros, as we like to call them. And the eventual goal for this project is to complete this heat map where we characterize all possible variants of uncertain drug resistance against all approved drugs, approved tyrosine kinase inhibitors against ABL. And the eventual goal is to help geneticists and clinicians help them make treatment decisions by providing them heat maps like these. So for the next 2 to 3 minutes, I'll pivot over to another cool finding that we saw when we compared our saturation mutagenesis data to base editor screens. So we're pivoting right now. So I talked about drug resistance so far, but now we're going to look at kinase function and kinase fitness. So BAF3s, as I mentioned, are a tool cell line that are growth factor independent when you transduce them with a functional oncogene. However, the worse the transforming potential of the oncogene, the worse the fitness of the BAF3s under this cytokine independent growth. So we did a screen for kinase function, both using saturation mutagenesis and base editing. Some of the base editing efforts were led by Ivan Sokerny, who is our in-house base editing expert. And I just want you to notice how wide the editing window of base editing is. So the orange area is the region we were able to mutagenize using saturation mutagenesis with a lot of troubleshooting. With base editing, you can do a lot more. You can even do multi-gene. So it's definitely, there's good things to be had with it because you can introduce endogenous variations and you can study things in the right genomic context. But when we compared base editing data to saturation mutagenesis data, most of these signals from base editing, if you look at just residue level data, were well correlated. So base editing was able to identify a major functionally consequential motifs. However, and you can see here, right, where we're looking at the catalytic domain, and if you look at the residue near the catalytic domain and mutagenize those, those are very functionally deleterious regions to mutagenize. And you see a stark difference going from red to blue as you go from 267 onwards. So base editing was able to capture that strong depletion signal. But when you look at more subtle cues, like signals that deplete and enrich and deplete and enrich, base editing, a lot of times it missed those signals, at least in our adenine base editor screens. And when we directly compared, when we plotted B and SM data against each other, we saw a modest correlations of the two. And the data clustered into 4 separate regions. And assuming that the saturation mutagenesis data is a ground truth, these are true negatives, these are true positives, these are false negatives by base editing, and these are false positives for depletion by base editing. And when we looked at which sgRNAs were making these mutants in the faulty regions, we realized that the sgRNAs that were making mutants in the true positive region were not making any other neutral mutants, if that makes sense. The sgRNAs in the false negative regime, on the other hand, were making not just the target mutant, but bystander mutants as well. And when we built a model, a mutant mixture model that uses saturation mutagenesis data to predict what the overall fitness effect of that mutant mixture would have been was made by the sgRNAs, those correlations matched the base editing data much better. So in the future, the goal of this project is to actually make these mutants in lab and actually deep sequence them with duplex sequencing and see exactly what percentage of the false negative signal is masked, is generated because of these neutral bystander variants. By our estimates, it's up to 40% of the true depletion signal is masked by these false negative variants, but I'll leave, we're gonna, before we draw any conclusions, wait on more data to come back there. So I'd like to acknowledge my lab for supporting me, specifically Justin and Josh and Marta and Ivan for all their help with the experiments, and I'll be happy to take any questions. Thank you.
Thank you so much for your talk. It's a very important research on pharmacogenetics or pharmacogenomics. And we already have questions in the Q&A. So William is asking that how can you tell more about how you converted the growth rates to depletion effects?
The growth rates to depletion effects. Yeah, so these depletion effects are basically essentially when the growth rates of the doubling time of the mutants is infinite, right? So it's pretty easy to see them. Maybe I don't understand the question correctly, but a normal doubling time of BAF3 cells is around 12 hours. And when you see that apparent doubling time increase to like 60 hours, we know there's a pretty strong depletion signal, as is evident by looking at the net growth rates of these mutants.
Okay, and I guess William has a question. He has his hand raised. You can unmute yourself and ask.
Okay, I was just wondering from technical, just a technical question with respect to the sequencing method. What do you think about the new PacBio systems and the Revio system? Is that a way to just like get away from doing this type of duplex sequencing? Because they estimate that you can do 1 in 100,000 or 1 in I don't remember, but yeah, they can reach quite low error rates. Do you think that if there's a future there for this type of studies?
Yeah, I mean, so PacBio systems have been started to be used in these deep mutation scanning screens. I can go over a lot of different things that are like better about duplex sequencing or about PacBio. But one of the things that comes to mind is that with a lot of these PacBio systems, you have to introduce barcodes at far distances from the actual variant, which is why you're able to sequence the barcode and the variant. And that is an extra cloning step in the library prep process, right? So with duplex sequencing, we don't have to do that. We can just have the mutant in our library and sequence the mutant directly, if that makes sense. So there's no extra cloning steps. And the other thing is with duplex sequencing, since we're going directly into the genomic DNA, we can use it for stuff like base editor screens, where if a mutant is present at 1 in 300,000, you can actually pretty accurately be able to look at its allele frequency and look at whether it's present in the pool or not. So those are the two things that come to mind, but not to say that those PacBio systems are not working well. They're clearly able to do some error correction there. Yeah.
Awesome, thank you.
We have one question from Mose. He's asking if you think you could pick up more subtle changes with prime editing rather than with base editing.
Yeah, that's the entire field is wondering about that question, right? Can we pick up more subtle changes? We can theoretically, with prime editing, mutagenize at a much more saturating way. You don't have the problem of bystander mutants. I think you could probably, honestly, you could probably pick up a lot of these subtle changes, because we think most of them base editing is not able to capture them because of bystander mutants. But if you don't have the problem of bystander mutants, I don't see why you wouldn't be able to pick them up. Maybe because with prime editing, because of the low editing efficiencies, you might not just know whether you're making a mutant or not. So that's the only place where I've not been sold on whether prime editing is ready yet, you know.
Thank you. We should be wrapping up, but I do have one more question that is kind of related to my research. So if you want to stick around for this.
Yes, for sure. Love to.
In one end, we have the resistance that you see variants that are in reach, but I'm wondering in the other end, if there's a threshold for variants that are so low and so depleted that could be correlated to toxicity. Because I know that some genes, an example of the gene that I work with, HXT, it's unrelated to cancer, but some studies has shown that if patients that have cancer take platinum-based therapy and have a minor allele mutation, they have less survival. So did you see any database or any validation of toxic variants that you could correlate with this DMS screening?
That's a good question. I think we looked at known deleterious variants, and a lot of those tracked with what we would expect. Like if we're just talking about how healthy the kinase is, the screen for kinase function, we know that hydrophobic residues are going to be deleted out of the popular, or deleterious, right? So we definitely saw that. But if you're talking about drug resistance, which variants are sensitized upon treatment with inhibitors, I don't think we specifically looked at that problem, but it would be an interesting thing to look at. I would just say though that sensitization is inherently a tougher signal to triage than resistance, because in resistance, your allele frequencies from baseline are enriching, and it's easy to tell apart when a mutant is going from 1 in 100,000 to 1 in 10,000. But if it's going from 1 in 100,000 to 1 in a million, then it might be tough to get actually accurate parameters.
Yeah, I imagine that would be a big challenge. So
Yeah.
thank you so much for your Q&A and your presentation. Thank you, William, for your presentation too. And thank you everybody else that stayed here. Oops.
Yes.
So just wrapping up, thank you everybody and see you next month.
Thanks, guys.
Thank you.